slides which is in that slide deck is kind of a mixture of some of the slides which are in lecture 2 and lecture 3 if you are looking at the slides on My Place. But when it comes to your exam and your multiple choice questions, whether you want to study the slides which are uploaded for the Strathclyde students on My Place, or whether you study the slides that I have gone through, so all the slides which are on the Strathclyde BM 108 My Place are also in the slide decks that we have gone through. But what you might have noticed is often in these slides that I have given you, there is more information, okay? So there's slightly more information, there might be slightly more text on some of them. And that is really just to help you study, because I know it can be sometimes be difficult to understand everything that's being said. So when it comes to studying for your assessment, it doesn't matter which slides you look at, because it should be the same information in all of the slides. It might just be in the slide deck that you have seen, there's extra slides, just to try and explain some of the things a little bit more to you guys, okay? Okay, so today, we're going to continue talking about innate and acquired immunity. And sorry, what I need to do first of all, of course, is to share the screen with you. You can't see it. Okay, so we should have shared screen now. Yeah, you can see the slides now? Okay, so
yes.
we're going to, yeah, you can see them good. So we're going to continue to talk about innate and acquired immunity today. Some of it might very well be repetition from yesterday, but it's really quite important that you understand how and which cells are involved in these two arms of the immune system. Okay, so these are the two different types of immune responses which are involved in your immune system responding to a pathogen. So you saw this one yesterday. There's the innate immune response and it's the acquired immune response. The innate immune response you're born with has a very quick response to any pathogens that might enter. It has no memory and it is not specific to any particular pathogens. This is unlike the acquired, or what we also call specific immune system, because the specific immune system has got specificity to pathogens. So once it has encountered a pathogen, if that same pathogen, if you're reinfected with that pathogen, it will remember that pathogen and you will get a very quick immune response. So the acquired immune response have got a memory. The first time you're exposed to a pathogen, the acquired immune response takes days to develop. So it's not as quick. It doesn't respond as quick as the innate immune system. However, once you have acquired immunity, once you have already been exposed to that pathogen and has got memory of it, the acquired immune response is very quick as well. Okay, so we'll start off with innate immunity. So I've already said it's non-specific. It obviously protects you against foreign cells or substances. Let's wait a wee second. There you go. Without having to recognize that particular pathogen specifically. It's an inherited ability. It doesn't require that you have previously been exposed to that invader or that pathogen. And what it does, it requires, it recognizes some general properties which are found on pathogens, such as some of the carbohydrates and lipids, which are present in the cell walls of many microbes. And this is what these cells of the innate immune system can recognize, and that's how they know that it is a foreign cell or substance which have entered the body. We spoke about the initial line of defense yesterday. Sorry? Somebody wants to say something? Hello? No? Okay, so I'll continue. So we spoke about the initial defense. So the initial defense of the innate immunity is the body surface. Obviously, microorganisms, very few, can penetrate the skin. Our skin also secretes molecules which are antibacterial. You know that we have got mucus secretes, many of our cells secrete mucus in the mucous membrane. So that's, for example, in the lining of the respiratory and upper gastrointestinal tract. And that is really, or that mucus can trap invaders. And it can also have antimicrobial activity. Also, you know, we have stomach acids, for example, and stomach acids can obviously destroy invaders. We talked about others yesterday. We're not going to go through them again today. What you really just need to know that body surfaces is the initial innate immunity response. However, if a pathogen is able to penetrate the barrier and gain entry into the internal tissues and fluids, there's other non-specific defense mechanisms which are activated. So it's important that recall listening to that I said non-specific defense mechanisms. Now, these defense mechanisms are mediated by the phagocytotic cells. So we talked a lot about these yesterday. We'll very briefly and very quickly go through them again today. So most of the phagocytotic cells belongs to the white blood cells, or the leukocytes, they're also called. There are 6 types: neutrophils, eosinophils, monocytes, macrophages, basophils, and natural killer cells. We went through all of these yesterday. The basophils does not have phagocytotic activity, neither does the natural killer cells. Natural killer cells can kill cells, and we'll get back to that a little bit later on. We actually spoke about it yesterday as well. The dendritic cells perform functions which are very similar to the macrophages. So they're antigen presenting cells. Remember, we talked a bit about yesterday about presenting antigen. The mast cells are very much involved in allergic responses, and the mast cells release histamine. We'll get back to that a little bit later on, actually. Now, so just a little reminder, what does it mean that a cell is able, or is a phagocytotic cells? Well, it basically means, in the example here, we're looking at the macrophages. So if you're looking at that schematic drawing here on the right-hand side, we just go through that. You can see the macrophages extends these pseudopodias, and that pseudopodia surrounds the bacteria, which is here in red. You get what is a phagosome formed, and it's obviously taken into the cell. Now that phagosome will fuse with what is called the lysosomes. Lysosomes contains enzymes, and these enzymes are able to digest the bacteria. Once the bacteria is digested, the material, as you can see here, is excreted via exocytosis. Now, what the macrophages is also able to do is through this process, it has got, we'll talk about that more later on. I'll just introduce the concept now. what it's also able to do is to complex this, some of these fragments from this pathogen, which what is called a major histopathologic complex. Now that complex, you will see later on, is really important for presenting antigen to other cells of the immune system. Now, I'm not going to go through this one again. We went through all the cells yesterday. So the leukocytes, the white blood cells, neutrophils, eosinophils, monocytes, macrophages, basophils, and natural killer cells. We went through what these were able to do yesterday. And we also talked about the dendritic cells and the mast cells. We also looked at yesterday how all the cells of the immune system has a common, is differentiated from a common hematopoietic stem cell. So there is a common multi-potential hematopoietic stem cell, and that differentiates into progenitor cells. So we have the myeloid progenitor cells, and we had the lymphoid progenitor cells. Now, the myeloid progenitor cells are the ones which are then differentiating to all of the white blood cells. On the other side, we have got the lymphoid progenitor cells, and these are the cells which can differentiate into the T and B lymphocytes, which are part of the adaptive immune system that we're going to talk later on. Now, it's also the progenitor cell to the natural killer cell is also down the lymphoid lineage, as you can see in the diagram here. Okay, so inflammation is an innate local response to an infection or injury. And what inflammation does is that it destroys or it inactivates foreign invaders, so pathogens, which gains access to intestinal fluid or tissues. Now, the inflammatory response not only destroys the pathogen, it also clears the area for dead cells, and then it sets the area up for tissue repair, because all this tissue obviously has to be repaired after it has been damaged. Now, the key cells which are involved in this process is the phagocytotic cells. So neutrophils, macrophages, dendritic cells, but also mast cells. And we'll see in a minute why the mast cells are also important. Now, what these cells are regulated and what their activation is induced by is by cytokines. So all these cells produce what is called cytokines. And these cytokines plays a very significant role in activating, not only activating, but actually also recruiting these cells to the site of where that injury has occurred. So let's look at an example here. So in this example, you have a tissue injury, as you can see here, which is caused by a splinter. So the splinter has induced or introduced bacteria beneath the skin. So there's bacteria in the splinter, and it has been introduced into the tissue through that splinter penetrating the skin. Now, the mast cells are very quickly recruited to the site, and they secrete a histamine. Now, the endothelial cells, which has obviously been injured by this or damaged by this splinter, they start to secrete nitric oxide. And those two are really important because it allows the blood vessel to dilate and they become really leaky. And because they become leaky, that now allows some of the other cells of the innate immune system, so the neutrophils, the macrophages, etc., to get entry into this injury site, so the tissue that is being injured. Now, the neutrophils, the cells which are first recruited into the site of injury, they produce cytokines, and it's these cytokines which are activating and recruiting other cells of the immune system. So the macrophages, for example, will be recruited because of cytokines which are being produced by these neutrophils. Now, the neutrophils and the macrophages, which have got phagocytotic activity, then will start to engulf and destroy these bacteria. Once that's done, the capillaries will turn to normal and the infection is obviously brought under control. So here's really just a summary of that slide. So the injured tissues, the mast cells and the neutrophils secretes these inflammatory signals. So these are cytokine that induces these vascular changes, that increases the blood flow to the area, and it increases it gives access for these cells, these immune cells, to enter the site of the injury. It also increases the permeability of plasma proteins to gain entry to the interstitial fluid. So once these phagocytes enter the area and they encounter these microbes, they release inflammatory mediators, and that brings in or activates even more phagocytotic cells to the site of that injury. Once the microbe has been phagocytosed, the fibroblast in the tissue will proliferate, start repairing the tissue, and obviously if that tissue repair has not occurred optimally, you can obviously get scar tissue. Okay, so in addition to phagocytosis and inflammation, as we've just looked at, the body also have a couple of other innate defenses. So innate, remember, it's not a specific defense against a certain pathogen. So it has two other innate defenses against pathogens without having previously been exposed to these. So for example, in response to viral infection, many cells produce what is called interferon, and they secrete this into the extracellular fluid. And interferon can actually inhibit, it binds to receptors on infected cells, and it can inhibit the viral replication of the virus in the host cells. Again, this is not, interferon is not specific to a particular virus. Another defense is the complement system. So complement is a range, it's a number of inactive proteins which circulate in the blood, and they become active by contact with the surface of microbes. So complements are different to phagocytotic cells. Complement proteins, they kill microbes without phagocytosis. And we'll look at this in a minute, but what they do is they use this membrane, or they launch this membrane attack complex. It creates a channel in the plasma membrane of the microbe, which obviously leads to the microbe bursting. Okay, so let's look a bit at the complement system. So complement is really circulating proteins which can actually kill pathogens through different mechanisms, and we'll look at these mechanisms here. So it can either kill, or so we just said it can kill directly, but it can also mark pathogens for phagocytosis. Now, the complement system consists of 25 proteins, and these proteins work really closely together, complement the action of immune cells in destroying bacteria. They circulate in the blood in an inactive form. Now we're going to talk a little bit about the different immune functions that these complements can trigger. So they can trigger phagocytosis, And they do that by opsonimizing antigens. Now, opsonimizing antigens really just means that they can label. So they can label pathogens with complement proteins, which then leads to phagocytosis. So the bacteria becomes marked by a complement protein, which is recognized by a phagocytotic cell. We'll look at an example in a minute. Also, they can induce inflammation, so they attract macrophages and neutrophils, and they obviously do that by marking. Again, as with the phagocytosis, what they can do is they can mark the bacteria or the pathogen with these complement proteins, which then attract macrophages and neutrophils, and they secrete all these cytokines, which leads to inflammation. They can also kill bacteria by a membrane attack, so rupture the cell wall of the bacteria. Now, here is an example of a complementary pathogen and how that activates phagocytosis. In this case, we're looking at a macrophage. So you see here in red a bacteria. So here is the bacteria, and that bacteria has been coated, so you can see, by complement C-3b. So this is one of the complement proteins. So you can see the bacteria, complement has tagged this bacteria with protein. Now, that complement protein can bind to this complement protein here. There's my, I couldn't find my marker. So, but here you can see that the complement which has been tagged on to the bacteria can bind to complement proteins on the membrane of the macrophage down here. I've lost it again. Oh, there it is. Which is called CR1 in this case. However, that does actually not activate phagocytosis. For phagocytosis to be activated, we need complement 5A. And complement 5A can bind to its receptor on the macrophage, as you can see here, and that will activate the macrophage to phagocytose the bacteria. So basically, the take-home message of this is that these complement proteins can tag bacteria so that they become recognized by macrophages. And then they facilitate that phagocytotic process. Okay, membrane attack. So they don't have to necessarily go through phagocytosis. They can also mark bacteria and attack the bacteria, kill them directly by forming this complex of proteins which is inserted into the membrane of the cell, and the cell then busts. So what you actually see here is the complement system facilitating or killing a bacteria that in this case has actually been tagged by an antibody. So you can see there's an antibody up here which is bound to an antigen on a bacteria or whatever pathogen this might be. And that has activated a whole cascade of different complements events, leading to the assembly of this cylinder membrane attack complex. So it consists of a different range of complement proteins, and that is inserted into the membrane of the cell. And obviously the cell will swell and it will burst because of external fluid entering into the cell and bursting it. However, pathogens can produce proteins that inhibit complement activation. And there is just an example of some of them here. I'm not going to go through all of them. But if you looked at this one here, meningitis pathogen, you can see that that can inhibit complement protein because it inactivates the binding of this C-3b. So this was the one we just looked at, which can bind to bacteria and then allow phagocytosis because it is recognized by macrophages. So there's other mechanisms, as you can see here. Okay, so you're probably sitting wondering, how does these immune cells recognize a pathogen? Well, they do that by recognizing specific molecules on the surface of the pathogen. Now, these are called pathogen-associated molecular patterns. These are patterns which can be recognized by immune cells. And the patterns are common to many types of pathogens. Now, how is that recognition accomplished? So how do they recognize and what do they do once they've seen these PAMs? Well, in 1985, a researcher, or two researchers, as you can see here, Wolfrath and Wieshaus, discovered that TOL1, which is a gene that is required for dorsal ventral development in the fruit flies and bacilli, also gave the flies the ability to fight off a fungal infection. So if the fly had TOL1, then they had an ability, or they were fighting off fungal infections. They later found that these TOL protein actually exist in all animals. They're found in the membranes of immune cells. So they're found in the membranes of macrophages, dendritic cells, and other immune cells, so these immune cells of the innate immune system. And what they do is they recognize and binds to ligands with these PAMs. So the PAMs, they consist of lipid polysaccharides. So these are the features which are found in the membrane of the pathogens. So it's lipopolysaccharides. It can be other microbial lipids and carbohydrates. It can be viral and bacterial nucleic acid, but it can also be the flagellum protein. So if you remember, some of the bacteria has got this flagellum, which allows them to move, and that can be that can be recognized by the immune cells. Now, these PAMs have very conserved molecular features, and the reason for that is that they're vital to the survival of the pathogen, so that is why they don't change. So when the TOL protein on an immune cell, so the immune cell has got the TOL protein, remember, when they bind to these PAM ligands, you get second messengers which are generated, and these are triggering cytokine secretions. Remember, it's the cytokines that stimulates the activity of other immune cells of the innate immune system. Cytokines actually also activate cells of the acquired immune response, and we will look at that later on. So most of these toll proteins are membrane-bound receptors, and they're typically referred to, you might have heard this, toll-like receptors. So it's these toll-like receptors on the leukocytes that recognizes the PAMPs ligands on the pathogens. Now that these toll receptors are very important for recognizing pathogens has also been shown when they looked at toll-4 mutant mice, so mice which are mutant for toll-4, and they showed that when they injected LPS, or lipid polysaccharide, which is one of the PAMPs which are expressed on bacteria. Obviously, when you inject that, it mimics a bacterial infection. And they found that if these mice didn't have the toll-4 or was mutant in the toll-4 receptor, then they were immunosuppressed. So they don't fight off the bacteria. Also similar proteins, so similar toll proteins has been found in plants. So it's thought that perhaps these toll-like receptors is one of the first immune defense mechanisms that evolved. So it's been around for a long time. Okay, so we're going to move on to talk about acquired immunity now. Obviously, unlike innate immunity, which is not specific, acquired immunity is specific. So it's specific to a pathogen. Cells of the immune system that first encounter a pathogen and later recognize a specific cell or protein to be attacked, as opposed to recognizing some general features on the pathogen. So it is not general features which is recognized by these cells. It is very specific features that belongs to only that pathogen that these cells can recognize. So it's not general, it's not PAMPs, it's not general features. It is very specific. They're also called antigens, very specific antigens, which can only be found on that very specific pathogen. Now, acquired immunity does require that you have been exposed to that foreign substance or to that pathogen before, because you only have acquired immunity to a pathogen if you have already been exposed to it. Now, an antigen, I've already mentioned antigen. So an antigen is really any molecule that can be found. This might be on a pathogen. This might be cells which are expressing antigen that is not recognized as self-antigen. So it is foreign antigen. So for example, in terms of cells which are virally infected start to produce different types of antigen which are foreign to the immune cells. Also, cancer cells, for example. Not all cancer is quite a complex topic when it comes to the immune system. However, many cancer cells express what we would call foreign antigen on the cell surface. Now, why cancer is still developed and are not picked up and killed by the immune system is because sometimes cancer cells can escape that recognition of immune cells, but that's a different topic. In most cases, actually, cancers are picked up by the immune system because they express this foreign antigen on their cell surface. Now, acquired immunity is found in all classes of vertebrates except for the jawless fish. It was originally thought to be absent in invertebrates. Okay, so we already looked at how the cells, we looked at the progenitors of these cells already earlier on. So these cells are also called lymphocytes. So I'm not going to go back because it's quite a few slides back actually, where we looked at the progenitors. So if you remember the lineage and the differentiation of these different cells. However, the cells which are responsible for acquired immunity, so the B cells and T cells, they are a type of leukocytes. They're also called lymphocytes. Okay, so lymphocytes. Now the lymphatic system, these lymphocytes, circulates in the blood, but most of them resides in organs of the lymphatic system. And we will look at in a minute what these organs are. So the lymphatic system has a network of lymphatic vessels, and those vessels drains the fluid, this is known as the lymph, from intestinal fluid, and it returns it to the fluid, to the blood plasma. On the next slide, we will look at the different lymphoid organs and tissues and where they're located throughout this lymphatic system. There's primary lymphatic organs, and this is where the lymphocytes are differentiating into mature immune cells, typically the bone marrow. Now these then supplied, these mature lymphocytes is then supplied or circulate to secondary lymphoid organs where they can differentiate. So what these lymphocytes do is they kind of circulate through these lymphoid organs, intestinal fluid, and then back to the lymphatic system. And they're continuously circulating that system. It obviously increases the chance of then encountering any pathogens. Right, so if you look at the figure here, you can see in red, we've got the primary lymphoid organs, the bone marrow. So most of these are really made in the bone marrow. They differentiate in the bone marrow, in here. This is where the hematopoietic stem cells are found. So differentiating into these mature lymphocytes in the bone marrow. And these are then supplied to these secondary lymphoid organs. So these are in green. You can see them in green here with tonsils, lymph nodes, spleen, etc. And then you can see the lymphoid vessels here, also in green, that these can circulate through. So they circulate through these lymphoid organ, intestinal fluid, and back to the lymphatic system and encounters antigen. Okay, so there's different kind of lymphocytes which are participating in coordinating this specific immune system response. We have got B cells, you can see them here on the right side. We have got B cells, we've got plasma cells, cytotoxic T cells, helper T cells, and natural killer cells. We're going to start off with talking about the B cells. So the B cell is a cell which mature in the bone marrow. The B cell can form these plasma cells, and it's these plasma cells that can synthesize and secrete antibodies. Now, I don't know if you remember yesterday, but we did talk about the humoral immune system, and we talked about the cell-mediated immune system. The B cells are involved in the humoral immunity. Now the T cells, they don't mature in the bone marrow, they mature in the thymus gland. And the cytotoxic T cells, they can directly kill cells which are infected, mutated or transplanted cells. Now, what that really means is the cytotoxic C cells can directly kill cells which are presenting antigen that is a foreign. Now, helper T cells are very important cells. They might not have the ability to kill cells or pathogens themselves, but they are very important in assisting in the activation of B cells and cytotoxic T cells. So helper T cells are absolutely crucial for the activation of both B cells and cytotoxic T cells. We are going to go through why that is, but one of the reasons is because of cytokine secretion. We'll look at that mechanisms in a couple of slides' time. And again, the T cells are involved in what we call cell-mediated immunity. So not humoral immunity, but cell-mediated immunity. Now, the natural killer cells are very similar to the T cells. However, they don't perform a specific action. They're involved actually both in innate and adaptive immunity. So we did talk about natural killer cells yesterday in innate immunity. So there's 3 stages to the acquired immune response. The first stage is recognition of antigen. So it needs to recognize antigen on these cells. So what do, these lymphocytes, is that they synthesize a type of membrane receptor that can bind to a specific antigen. So basically lymphocytes have got receptors. These receptors, which we'll talk about later on again, are specific to a specific antigen. So it's not something which is expressed on all pathogens that these are recognizing. It is something which is very specific to one type of pathogen. And then you need to get activation. So we have recognition of antigen. We need to get activation of the lymphocytes. And then obviously we need to get some kind of attack against this antigen to get rid of whatever pathogen that it might be. Okay, so this is really just shown schematically here. So if you encounter a pathogen, something very specific antigen on that pathogen is recognized by these 3 cell types, so cytotoxic C cells, helper T cells, and B cells. The helper T cells secrete cytokines, and it's actually these cytokines which are activating B cells and cytotoxic C cells to proliferate. And we get what is called a clonal expansion. So does anybody know what clonal expansion means? No? Well, so clonal expansion, it's actually quite an important word in this scenario. And the reason for that is that the clonal expansion means that you get proliferation of cells, so B cells and T cells in this case, which have got that receptor which recognize, so this receptor which is very specific to that particular antigen that was recognized. So all the cells have got the same receptor, and that receptor is specific against the antigen which led to the whole activation of this system. Now the B cells will secrete into plasma cells, and these plasma cells will then make antibodies, and these antibodies can bind or are made specifically against that antigen. So they can bind to that antigen. And then there's different mechanisms, which we'll talk about in a couple of minutes, as to how the cell or the pathogen is then killed. Now, what also happens is that we get what's called memory B cells produced. So these memory B cells are cells which, if you encounter this antigen again, these memory B cells will very quickly proliferate and and then find or search out this antigen. Cytotoxic C cells is the same. So the cytotoxic C cells then do not make antibody, but they actually directly attack antigen-bearing cells, so cells which have got this antigen. So direct attack. The B cells makes antibodies which binds to the antigen. Again, we can get memory T cells made, and memory T cells basically means that if once your body has got rid of this infection and it ever encounters this antigen again, you will very quickly get proliferation of these memory cells, and they will get rid of that antigen or pathogen very, very quickly. So in most instances, you will not get ill. Okay, so stage 1, recognition of antigen. So during development, each of these lymphocytes synthesizes a membrane receptor. And that membrane receptor, as I can say, have already said, can bind to specific antigens. Now, antigens that binds to the receptor are then recognized by the lymphocytes. And it's very important to remember that each lymphocyte is specific just for one type of antigen. Activation, so the lymphocytes bind to the antigen, they will undergo cell division, and we will get clones. We've already talked about that in the schematic. Now, for the B cells, we get plasma cells, and these plasma cells then makes antibody, and it's those antibodies which attacks the antigen, and you get memory cells made for future attacks. Attack against the antigen. So the cells differentiate into plasma cells, they make antibody, antibody is recruited, and so these antibody binds to the antigen, and that recruits. So we actually already looked at how the antibody can recruit the complement system, for example. So it can lead to activation of the complement system, and the complement system makes this cylinder which is inserted into the membrane of the cell. So it's actually the complement protein which is killing the cell, but it is the antibody which is bound to the antigen that is triggering this process of killing or attack. Activated cytotoxic C cells, on the other hand, as I've already said, carries out that attack themselves. So they've got protein called perforin, and again, perforin can be inserted into the membrane and kill the cell. Now, once the immune system has got rid of antigen, this is quite important actually, these plasma cells and cytotoxic T cells, except for the memory ones, of course, the memory ones will not die, but the cells which are actually performing the attack on the antigen, they will die by apoptosis. And that's obviously very important because it prevents an excessive immune response. And many of you might very well be aware of some of the diseases which are associated with excessive immune response. So autoimmune diseases is what this is leading to, such as multiple sclerosis, which is just one example. Rheumatoid arthritis is another example. So a whole range of autoimmune diseases, which are really because the immune system is not switching off. Now, the memory cells, as I said, are persisting because these are the ones which will very quickly respond to any invaders that has got this antigen presence. So any invaders where this antigen is present will be attacked very quickly because these memory B cells can very quickly proliferate. Okay, so we've got to go and look a little bit at the mechanism of how these are activated. So B cells and humoral immunity we'll start with. So as I've already mentioned, helper T cells are very important because it's the helper T cells which activates these B cells. So the B cells are activated not only by the antigen, but they also do need helper T cells for their activation. And this is because the helper T cells are secreting cytokines, which leads to the proliferation of the B cells. So proliferation into plasma cells that then makes antibodies. Obviously, the humoral immunity is a major defense against bacteria, viruses, and other microbes in the extracellular fluid. Okay, so B cells, humoral immunity, really important. We saw that yesterday in one of the diagrams, how humoral immunity really is for attacking or defending against bacteria and viruses which are in the extracellular fluid. So not intercellular, which is really where the T cells comes in. Okay, so I've already mentioned that these that the B cells makes antibodies. Now these antibodies, they're also called immunoglobulins. These antibodies are similar to the B cell receptor. So the receptor on the B cell, which is recognizing that antigen in the first place, the antibody that is produced is very similar or is similar to the receptor. There's obviously a transmembrane domain in the B cell receptor, which you obviously don't have in the antibody because it's not, it's circulating the antibody. Okay, so these immunoglobulins is composed of 4 interlinked polypeptides, 2 long heavy chains and 2 short light chains. There's 5 classes of immunoglobulins: IgM, IgG, IgGE, IgGA, and IgGD. Everybody, all vertebrates, have got IgGM. So IgM and IgG are the most abundant ones in mammals. So they really provide the bulk of specific immunity against bacteria and viruses in extracellular fluid. You can see up here that the IgM is a pentamer, see that here, and the IgG is a monomer, as you can see over here. Now, IgE, again, you can see that here, it's a monomer, and it participates in defenses against multicellular parasites. And it's also really important in allergic responses. IgA, secreted by plasma cells in the lining of the gastrointestinal tract, the respiratory tract, and the urinary tract. So it acts very locally. It's also one of the major antibodies which is found in milk. So it can be secreted in breast milk. Now, IgA is a dimer, as you can see down here. Finally, IgD, which is shown down here, is a monomer. The function of IgD is much more unclear. So you can see here that that's the B cell receptor on the left side and the antibody on the right side. You can see that the antibody and the receptor consist of a constant region. So this region is similar amongst each class. So it's the same for all of the IgM antibodies. It's the same for all of the IgE antibodies, etc. We then got a light change region up here. And importantly, we've got this variable region up here. So that variable region up here is what is varying depending on the antigen. So that's where you get the specificity of the antibody, because that only allows a very specific antigen to bind to the antibody or indeed to the receptor. You can see that the only difference really between the receptor and the secreted antibody is, as I said previously, this transmembrane domain that we've got down here, because obviously the receptor is inserted into the plasma membrane of the B cell. Okay, so the B cells, when activated, will then divide into plasma cells and memory cells, and then we get what we call clonal expansion. Already talked to you about what clonal expansion is. So we'll just jump over that one. Now, when it then comes to once the B cell is activated and plasma cells and antibody is made, how is the antigen or the pathogen which is expressing the antigen actually killed? Well, the antibody can inactivate the pathogen through a number of different mechanisms, as you can see down here. So we've got the opsonization, which actually we've already talked about. So this is really where the antibody is marking the cell for other. So for example, that can be for complement proteins to come in and destroy the cell, or it can be for other immune cells to trigger an attack, so macrophages, etc. What it also can do, these antibodies can bind and block specific sites which a virus, for example, is requiring for entry into a cell. So it might be that the virus no longer can be taken up into a cell because the binding site has been blocked by a pathogen. Here is just some examples of how B lymphocytes and antibodies neutralize pathogen in different conditions. So for example, poliovirus, the antibody prevents the virus from uncoating. So if it doesn't uncoat, it can't proliferate. It can also prevent the virus from transcribing in the genome. And that's, for example, how the influenza virus can be stopped. The last one I've already talked about, and that's receptors on the outside of the cell. So if an antibody is blocking the receptor through which the virus is taken up into the host cell, obviously we can't get viral replication. So we've already talked about that the antibodies are divided into these 5 major classes based on their constant region. So it's the constant region which decides what class you belong to, but also based on their immune function. It's the constant region which really determines the mechanism which is used to destroy the antigen. You can see that here in the figure. So if you look at the functional activity, So you could see that neutralization, for example, the IgA is very good and uses that mechanism a lot, while, for example, IgD do not neutralize, does not use that mechanism to destroy antigens at all. You can also see that some are better at activating the complement, for example. So if you belong to the class IgM, you're very good at activating the complement system, and it's a mechanism which is used to destroy antigen. Well, for example, if you belong to IgA, that mechanism is not the main mechanism which is used to destroy the antigen. Now, if you look down at the bottom of that figure, so down here, you can see that these antibodies also got different abilities. You can see, first of all, that the most common one in serum is the IgG1, so it's in at the highest concentration, as you can see here. Some of these antibodies can transport across the placenta, so the IgGs, many of the IgGs can be transported across the placenta. Some can be transported across epithelium, so IgA, as you can see here, etc. Okay, so this is really just showing you how antibodies can tag microbes for phagocytosis. So we have got, as you can see here, a pathogen, and on that pathogen, we have got antigens expressed, which is recognized by the IgG antibody, as you can see here. Now, so the phagocyte, so in this case a macrophage or a neutrophil, they express receptors for this constant. So we just talked about how they have this constant region. You can see that constant region is obviously there. The specific region of the receptors up here, because that is what is recognizing this antigen. However, that common region down here on the receptor, on the antibody, that is recognized by receptors on phagocytotic cells. The macrophages and some of the neutrophils are expressing receptors, Fc receptors, which can bind to that constant region. So they bind to that constant region, leads to activation, and we get a phagocytosis of the microbe. So basically the take-home message is here that the pathogen has been labeled by antibodies. And it's these antibodies which, by binding to receptors on the phagocytes, activates the cell and leads to phagocytosis. So basically the antibody is labeling the cell for phagocytosis. Okay, so very much is what is shown here as well, actually. So we have an antigen which binds to the B cell, which is displaying a specific immunoglobulin. And the binding of that antigen stimulates only cell division of that particular B cell. So the B cell which have got the receptor that can bind that antigen. So we get clonal expansion. After clonal expansion, we get plasma cells, and these plasma cells starts to make antibodies. Now these antibodies recognize the original antigen, so the antigen that the B cell bound to. Some of these differentiate, some of the plasma cells, or some of the B cells, sorry, will differentiate into memory cells. And it is these memory cells, in the event that you're ever exposed to this pathogen again, that will very quickly proliferate and very quickly eliminate that particular pathogen which is expressing this very specific antigen. Okay, so we're quickly moving on to cell-mediated immunity. Again, like the B cells, the T cells have got receptors for antigens, and these are very specific to very certain antigens. Okay, so the receptor is in the plasma membrane of the T cells. It's not secreted like immunoglobulins, so we don't get antibodies made. Now, during DNA, during development or maturity of these T cells, you get what is called, don't want you to focus too much on this because you will hear about that in later years, Willie, but you get multiple DNA rearrangements, which means that you get millions of different distinct types of these T cells receptors, which is therefore very unique to one specific antigen. We're not going to go through the whole mechanism behind these multiple DNA rearrangement and how both T cells and B cells are able to express this huge variety of different receptors, so that they can recognize these very they can recognize antigens very specific. However, unlike B cells, T cells cannot bind to antigen unless that antigen is complexed with MHC, class 1 receptor. Now, this is quite important, so you need to remember that it can only bind if the antigen is complex with what is called the MHC class 1 receptor. We'll get back to that later on. This is just really showing you. So we've got, in this case, a T helper cell. And you can see that it's got a receptor, transmembrane receptor. Again, we've got a constant region, we've got a variable region, and the antigen obviously binds to that variable region. Okay, so what is the MHC complex, so major histocompatibility complex? It's really an identity tag. So MHC1 is really found on all cells in the body. Now, the MHC can be complexed with antigen, and that is how an immune cell knows that there is something wrong with this cell because it's not expressing the normal MHC1 tag. Now, there is another class of MHC, and that is MHC class 2. Now, unlike the class 1, the class 2 is found only on antigen-presenting cells. These include macrophages, B cells, and dendritic cells. And T cells binds to class 2 MHC. Okay, so we're going to look a little bit closer at that now. So how do these foreign antigens become complex with these MHC proteins on the surface of the body's own cells? Well, they do this through antigen-presenting cells. So we've already said that helper T cells require class 2 MHC to function. And macrophages, B cells and dendritic cells, they present antigen with the class 2 MHC complex. So helper T cells can recognize and bind MHC complex, which is presented by B cells, dendritic cells, and macrophages. That then means, well, that is present, so the MHC class 2 complex is presented on phagocytotic cells through digestion of the pathogen. So the pathogen is digested into fragments or epitopes, and these epitopes can then become complexed with these MHC proteins. That complex is then transported to the cell surface of the macrophages, for example, in this case, and the T cells can then bind to that complex. They use the CD4 protein linking to help with that. Now, essential to T cell activation is also the secretion of cytokines. So cytokines help stimulate T cell proliferation. Now, it's much easier to understand when you look at the figure here, actually. So we've got a macrophage here. So the macrophage, as you can see, is phagocytosing an antigen. So it's come across this antigen and it's phagocytosing it. Now, what happens in the endosome of the cell, as you can see here, that fragments of that pathogen is complexed with the MHC2 class protein complex here. That is then transported to the surface and displayed on the surface of the macrophage here. Now, a helper T cell can recognize and bind to that MHC complex, which is presenting this antigen. And that occurs through this helper or this CD4 binding here is also required. It does require co-stimulation as well, but we are actually not going to talk about the co-stimulation today. Now you get secretion of IL-1 and TNF-alpha from the macrophage. So remember, these are cytokines. Remember, we talked about how cytokines are really important in stimulating other cells. What happens is that as these are then secreted, this is stimulating these helper T cells. And these helper T cells can then stimulate B cells and cytotoxic T cells. And we will look at that a little bit closer in a minute. So cytotoxic T cells, as I already mentioned earlier on, requires presentation of these class 1 MHCs. So remember, these are MHCs, protein complexes, which are expressed by all nucleated cells. And therefore, it's the same, we talked about it yesterday with the natural killer cells, because actually the natural killer cells can detect when cells are not expressing normal MHC1 protein. So it's when it's being complex with antigens that these cells, both natural killer cells actually, and cytotoxic T cells, can pick up these antigens. So virus-infected cells and cancer cells very often express these unusual proteins, which are fragmented with this MHC1 complex. And you then get presentation on the cell membrane of an MHC1 complex, which has been complexed with these unusual proteins that are made by virus infected and cancerous cells. Now, the cytotoxic T cells can then bind to that complex, and that releases chemicals that can kill infected or cancerous cells. So perforin, as I mentioned earlier on, and actually it's mentioned on this slide here, is one of the ones, the proteins, which is released by T cells and can kill the infected cells. So very quickly recap of what I've actually already said. So the role of the cytotoxic C cells. So virus-infected cells produces foreign protein, which is placed on the host plasma membrane, complex with this MHC1 protein. The cytotoxic C cell can bind to that antigen, and it can activate helper T cells to release cytokines, which are then stimulating the T cells to proliferate, and it leads to the cytotoxic C cells releasing proteins such as perforin, for example, which can kill the cell. This here is really just showing the cytotoxic C cells, which is bound to the surface of virus-infected cells. So you can see this cells here. This cell here is presenting antigen on its surface, which is complex to MHC1, recognized by the T cell, and it binds. Okay, so as I already mentioned, and as we've talked about, just because the cytotoxic C cells is bound to the surface of this virus-infected cells here, it does not mean that it's activated. So the cell needs the T helper cell for its activation. Now, how does these cytotoxic C cells get activated by the T helper cells? Well, that's what we're seeing here. So we've got a cytotoxic C cells, which is bound to the surface of this virus-infected cells because it has recognized this class 1 MHC protein, which is being complex with the antigen. So that's what it's binding to. This cell is presenting antigen to the cytotoxic C cell. Now, the helper cell, so we looked at that earlier on, and it's just really the figure that you see here. So here we have got a T helper cell, which has recognized the class 2 MHC protein, which is expressed on this macrophage here. This macrophage has picked up the same pathogen and antigen, which is expressed by the virus-infected cells. And it is displaying that on the surface of the cell here. The T cell is binding to the class 2 MHC, which is presented by the macrophage. So it's an antigen-presenting cell. Presenting it to the T cell binds, it proliferates, and, I should have said that the macrophage make these cytokines. It's proliferation of the T helper cell. The T helper cell secretes cytokines, IL-2, for example, and other cytokines, and these cytokines then stimulate both the T helper cell, actually, and the cytotoxic T cell to divide. So basically, we get activation of both of these cells. Okay, so really we've got the very same at the top of this figure here. So we have got our T cell, cytotoxic T cell, which is bound to the cell which is expressing the virus. And we have got our helper T cell here, which is bound to MHC2 complex, which has been complex to the same viral antigen as the one which is presented by or being picked up by the T cell here. So we get stimulation and proliferation of these cells. The cytotoxic T cells, so they proliferate and they'll bind to other virus-infected cells. And as I've already said, perforin is one of the proteins. It also secretes other proteases, but perforin is one of the proteins, and perforin is actually inserted into the membrane of the target cell, of the virus-infected cell, and obviously that leads water to be taken up into the cell and therefore burst. So the cell is now dead. Now, we already talked about something similar like this yesterday, but just to say that the natural killer cell destroy virus and cancers infected cells by secreting toxic chemicals, and it actually, like the T cell, can kill virus infected or cancerous cells, but it does that by recognizing general features on virus infected cells or cancerous cells. So it's not a specific antigen that is recognized by the natural killer cells. Okay, so here is a summary of the humoral immune response. So it's now putting everything together. So again, let's take an example. We've got a bacteria which enter the intestinal fluid through a wound. That's what you're seeing up here. Where's my, it's gone again. Oh, so that's what you're seeing up here, okay? Got a bacteria which enter the fluid through a wound. That bacteria then is carried into the lymph node. And in the lymph node, it binds to macrophages and B cells. Now, the macrophages, we know it's a phagocytotic cell. So the macrophage is phagocytosing the bacteria, and it is complexing part of that bacteria with the MHC protein complex. And that, I've lost it again there, that MHC protein complex with that processed antigen is then presented here on the surface of the macrophage. Now that can be recognized by a T helper cell. The T helper cell can recognize that MHC class 2 protein with the antigen on it. So the T cell binds. As I've already said, we need CD4, and we also need co-stimulation here. We're not going to talk too much about those today. We're not going to talk about them at all today, basically. So the macrophage is also secreting cytokines, IL-1 and TNF-alpha primarily, which stimulates the T helper cells, and it starts secreting IL-2 and other cytokines, and that leads to this T cells starting to divide. Now the T cell can bind to the B cell, which is displaying, so the B cell is displaying the same antigen on its surface as was presented by the macrophage. Now that leads to proliferation because of the cytokines produced by the T cell. We now get proliferation of these B cells, we get plasma cells, and we get secretion of these specific antibodies, which can then bind and lead to destruction in different ways of the bacterial cells. Okay, so I'm going to talk a little bit about immune tolerance. So how does the body distinguish between self and non-self? How does it know when it's foreign and when it's not? We have talked a little bit about that already. So there's quite a large diversity of lymphocyte receptors. And they also generate receptor binding to self. It's two mechanisms which are preventing an attack on the body's own cells. And one is called clonal deletion and one is called clonal inactivation. Clonal deletion occurs during early development. So basically it involves T cells in the thymus, which are responding to self-antigens being destroyed by apoptosis. Clonal inactivation occurs outside of the thymus, and it potentially self-reacting T cells becomes non-responsive. B cells undergo a similar process to this. Now, autoimmune disease, I have already alluded to it. So autoimmune disease, this is when the immune system attacks the body's own tissues and cells. Examples is multiple sclerosis. So in multiple sclerosis, you get attack on the myelin sheets, which are wrapped around the neurons and are involved in neural transmission. Myasthenia gravis is a disease where the acetylcholine receptors, which is found on skeletal muscles, are attacked by the immune system. Rheumatoid arthritis is inflammation, particularly in the joints. Type 1 diabetes is an autoimmune disease where the immune cells have destroyed insulin-producing cells. Now, there really isn't any cure for any of these diseases. There is treatment options, so we're treating the symptoms of the disease, and they obviously typically involve suppressing the immune system. Except for type 1 diabetes, you're very often treating the patient with insulin. But most of these involves suppressing the immune system. We're going to talk a little bit about immunological memory. We already talked a little bit about it yesterday. So the acquired immune response to a given antigen really depends on previous exposure. So the primary immune response is the initial exposure to an antigen. And in that response, in the primary response, the antigen production is much slower, and it is much slower than what you see in a secondary immune response. So that is the reason why you might, the first time you're infected with a virus, for example, you might get ill as the immune system is fighting the virus. But the second time, and if you're exposed to the same one again, you get a very fast and a very high antibody production, which means that you very often will have no symptoms. So your immune system is very quickly getting rid of it before it actually develops into disease. And this is because of the memory B cells. So the memory B cells are very quickly stimulated to differentiate and multiply. And that's really just what is shown here. So this is the antibody response in primary and secondary immune infections. So you can see here your primary response takes weeks to develop, and the amount of antibody that is produced is much lower than what you see in the secondary immune response. So in secondary immune response, a second exposure, very high titer of antibody produced very, very quickly. Obviously, that is what you're exploiting with vaccination. So with vaccination, what you do is it can be different things. We're not going to talk too much about that today, actually, but what you inject the person with could be a live or a weakened form of a pathogen. It could be an inactivated pathogen, or it can actually even be purified of recombinant protein. But what you are aiming for is injecting something or administering something to that individual which will lead to an immune response. So you're looking for an immune response because you need an immune response to make these memory B cells. Okay, so active immunity versus passive immunity. So active immunity is when you have acquired a response to an antigen, so you have been exposed to it, you've got memory B cells, and these memory B cells, it very quickly eliminates the pathogen or whatever is expressing this antigen. So that could obviously be you being exposed to this naturally, or it could be that you have been vaccinated or immunized against whatever this antigen or pathogen might be. Now, passive immunity is different to active immunity, because passive immunity means that you have had transfer of antibodies from one individual to another. So, for example, newborn babies can be protected against some diseases when they're first born, and that is because of transfers from the antibody from the mother into the child through the breast milk. So your child will get some antibodies through breast milk, which means that they do have some form of immunity when they're first born and receives breast milk. It could also be, for example, if somebody is administered or injected with antibodies that has been isolated from somebody else. It is very short-lived, this, because you don't produce memory B cells in this scenario. So passive immunity does not lead to the production of memory B cells. And it's obviously only when you have got memory B cells that you have got active immunity. Okay, so we are going to finish off the lecture talking a little bit about the impact of the immune system, or what role the immune system plays in a couple of different diseases. So one of the biggest contributors or one of the biggest factors that can affect the immune system is lifestyle and nutrition. So protein, if you have malnutrition of calories or proteins, you can decrease your resistance to infections. And the reason for that is obviously that you need amino acids to synthesize essential proteins for the immune system. Otherwise, it becomes impaired. Now, cortisol is associated with stress, and it has a very powerful suppressive activity on inflammation and specific immunity. So high cortisol levels is typically associated with stress. So cortical secretion, so hormone which is increasing with stress. Now, physical exercise can also have an effect on your immune system. So moderate exercise is good. However, very strenuous physical exercise is actually not thought to be beneficial for you. And the reason for that is that it increases tissue stress. And that leads to immunosuppression. Now, you might have guessed this, but obviously transplant and transfusions. Immune cells are really important because they lead to rejection of transplant. So there's 27,000 organs transplanted in the United States every year, and graft rejection is one of the major problems. So why do you get graft rejection? Well, the immune cells which are involved in it is the cytotoxic C cells, and it is the helper T cells. And why is it? Well, it is because the MHC proteins, which are expressed by individuals, are not the same, except for in identical twins. So the MHC class II proteins differ from individual to individual, which means that if you get a graft transplant, the immune system is going to recognize these cells as being foreign, so foreign antigen, and you get activation of the patient's immune system, and it will start attacking and destroying the new organ. Now, how do you circumvent this? Well, what you probably are aware of and might have heard of is this patient is given immune suppressive drops after they have received a transplant. Now, the immune system also plays a massive role in blood transfusion. And this is antibodies which are involved. So you will all know that our red blood cells, so you probably all know about the blood type system. So you all know that on our red blood system, on our red blood cells, they're also called erythrocytes, the red blood cells. But the red blood cells have got antigens on them. And you will know that if you have got a blood type A, your red blood cells will express antigen A. But what you also do have is then antibodies against the other antigens. So you're expressing antigen A on your red blood cells, but you have got antibodies against antigen B. So that is what you can actually see down here. If you have got a blood type B, you've got antigen B in your red blood cells, and you have got antibody A in your plasma. Group A and B, so if you're AB blood type, you don't have any antibodies in your plasma. If you're group O, you have obviously got no antigen on your red blood cells, but you've got antibodies against A and B circulating in the plasma. So blood transfusion, you need to think very carefully about what blood type is the one receiving, the recipient of the blood, and what is the blood type of the donor. The reason for that is, so let's take an example. If you are the recipient, you are blood type A, you have antibodies in your plasma against antigen B. Therefore, if somebody with a blood type A was to receive blood from somebody that has got a blood type B, the antibody in the recipient would obviously bind to the antigen on the red blood cells, and it would lead to destruction by the immune system of these cells, which means that somebody with a blood type B could not donate blood to somebody with a blood type A. So basically, you could, somebody with a blood type A could only receive blood from group O because it doesn't have any antigens on the surface of the cell. Again, if it was somebody with a blood type AB that was donating, those cells would be destroyed because the antibody which is found in the recipient would basically destroy the red blood cells received from the donor. Okay, so we are very quickly going to talk about allergies. Allergies are also called hypersensitivity. This is basically where an immune response is triggered to environmental antigens, and that leads to inflammation and damage to cells in the body. Allergens are basically antigen cause that is causing allergies. So we call them allergens, and it is basically antigens which are causing allergies. Now, most of these allergens are pretty harmless. It's how the immune response is responding to them that is really the problem. So allergies are classified according to the speed of the reaction. So we've got delayed hypersensitivity. It's a response that takes several days to develop. And you see it, for example, I don't know if you know the plant poison ivy. but people which can often be allergic to this plant, and it generates a rash, but that takes several days to develop. Immediate hypersensitivity is much more common, and it's actually caused by IgG antibodies, which attach themselves to mast cells, and it leads to release of inflammatory mediators versus histamine, and that induces an inflammatory response. As you might know, people with allergies, and you might know that that induces mucus secretion, increased blood flow, etc. And you get swelling of the epithelial lining, and you can get contraction. So some people might actually find it difficult to breathe if they have a really severe attack. It also produces a runny nose, sneezing, and obviously difficult breathing because of that contraction. Okay, the last one we're going to quickly talk about is HIV or AIDS, which can lead to AIDS. So it's also called acquired immune deficiency syndrome. It was first described in 1981, and it is caused by the human immunodeficiency virus, so the HIV virus. And what the HIV virus does, it infects the helper T cells. So the cytotoxic T cells actually kills these infected helper T cells. So without adequate helper T cells, neither B cells or cytotoxic C cells can obviously function normally, because we've just talked about how these helper T cells are really important in activating B cells and cytotoxic C cells. Now, people that die from infection with this virus is very often actually not because they die of this particular virus, but it's really because of other infections that their immune system is not able to fight because it is suppressed. So they are immunosuppressed because they don't have a normal functioning immune system. So when they're picking up other infections, they're not able to fight these infections off. There's 15,000 new infections every day with this virus. And this is just showing you a figure of the people which are living with HIV worldwide. You can see there was quite an increase in the 1980s, but it's leveled out very much from 2000 and onwards. So the majority of individuals which is infected with HIV show no signs of AIDS. They're diagnosed really just by the presence of HIV RNA in the blood. But if the patient is not treated, then AIDS tend to develop within a time frame of about 10 years. Initially, your body is replacing these helper T cells by new cells. So because the cytotoxic T cell is killing them, they're easily replaced. However, over subsequent years, what happens is that these helper T cells starts to decline in their concentration. Okay, so that was a long lecture today. So tomorrow we have got a shorter lecture. There is very few modifications to the slides tomorrow, but I will send you the slides that we're looking at tomorrow. So it's a much shorter lecture than today. And what we're also going to do tomorrow is that we are going to go through all of these MCQs that we have at the end of each of these lecture slides, okay? But as I've already told you the other day, I am not going to be the one who answers these questions. You are going to have to answer these questions. Okay?
Okay.
So you have to speak tomorrow.
Okay.
Right? So no, tomorrow
Right.
it is talking day. Yeah. So you better get your talking practice tonight and get your English going, okay?
All
Right. Okay, have a nice evening time. It must be in China now, so have a nice evening, guys. And I'll see you all tomorrow, okay? Have you got any questions or anything you want to ask me before we finish? No? We're not getting the talking today, but tomorrow, remember to bring the talking along for tomorrow. Yeah? Okay, I'll see you all tomorrow. Bye-bye.
Bye.
Bye.